Hello, and welcome to the webinar. This is me. I won't introduce myself because I think someone else is going to, but I'm the product manager at Horizon Diagnostics, and I've been here for 3 years. At Horizon Diagnostics, we're doing a number of very important things in the world of molecular diagnostics, where the goal and aim is to develop reference standards that will help to support the understanding of the limit of detection of particular assays, as well as the robustness of their workflow. What I'm going to be talking to, together with my colleague Hadas, is very much of how formalin can impact clinical diagnostics, which is becoming more and more important and coming to the forefront of people's questioning on how their assay is performing. I tend to always like to start my my webinars or presentations by really pushing this question out there to the audience, to you guys in the audience, and it is: what is the impact of assay failure in your laboratory, and how do you monitor for it? And it goes on to beyond that to what happens if it does go wrong? How can you then mitigate for those times when it can go pear-shaped? Now, if you look at the application of companion diagnostics, I've put together a very simple workflow here, where you have your cancer patients. They then undergo surgery where you're cutting them open. You take a biopsy. You then process that biopsy to generate a FFP sample. At this point, you're treating it with formalin. You then have to do a DNA extraction step. For me, this is where, together with the FFP sample processing and the DNA extraction, I would call this the pre-analytical step, and then you go on to the diagnostic assay, which will be the analytical step. And all of this together within companion diagnostics is looking to drive better treatment for cancer patients, because you're looking to triage patients where if they have a specific mutation, you can push them onto a specific and positive drug therapy. If you break that each step down a little bit more to a granular level, you then have on the pre-analytical side, you have your biopsy. It goes through this FFP processing, organ extraction, storage. You then have the DNA quantification at the end point, which is critical to then understand how good the DNA is coming out of your assay, and also how much you then have. On the flip side, on the analytical side, you've got your DNA sample, which you've quantified with whether it's a nano drop or a fluorescence-based method. You then have to do some sample preparation. It could be a qPCR diagnostic assay you're running. It could be Sanger sequencing based. It could be next generation sequencing, which is becoming more and more adopted into molecular diagnostic assays. And the output from that is then your actionable decision. All of this together, and the analytical side of the process will govern really the quality of your diagnostic result. So bringing this all around, and to bring it on the topic of this webinar, you have at the start, when you, in the pre-analytical step, and I'll try and use the mouse so you can see where I'm talking. As soon as you take your sample from your patients, as soon as you've opened them up and they're under surgery, that sample will be undergoing various oxidative damage. And that can then potentially induce apoptosis, or could go through necrosis. You're then going through the formalin treatment to generate your FFP blocks. Now, formalin is very good at preserving samples, but it's also an excellent DNA damaging agent and a pretty nasty chemical. And so, in this step here, whilst you're fixing your sample and preserving it, you're also introducing potential cross-links in the DNA, potential fragmentation, as well as oxidative and methylation onto the DNA. Through the process, you then do DNA extraction, and then you have your DNA at the end point. And like I said, it could be highly cross-linked, it could be highly fragmented, could have oxidative damage, or could be methylated. And the extent of these modifications is governed very much by how long the sample has been either hanging about pre-formalin treatment or how long it's actually undergone formalin treatment. This will then be fed into the DNA quantification. Depending on the quality of the quality of the DNA and the formalin treatment, the DNA extraction could be impacted where you have a decreased DNA yield and also a decreased DNA quality. All of this together and the quality of the DNA could actually lead to quantification errors, where you actually start to, as you increase the fragmentation of the DNA sample, you can actually start to over-quantify the amount of DNA you have in a tube. That will then lead on to the analytical step, and it very much comes back to, if you've got high quality DNA, then you can do the analytical step very well. If you've actually got DNA that isn't so clean, isn't such high quality, you're going to start to struggle to actually initially quantify it, and then you'll be testing your assay sensitivity and your assay specificity, because if you don't put enough DNA in because you've over-quantified, then you're actually going to be really pushing the limit of detection of your assay. And this can potentially lead to either the occurrence of false positive mutations from the analytical side of your assay, or false negative mutations, and ultimately lead to an inaccurate and actual decision. So that's kind of a nice overview of what where formalin fits into the workflow and how it can contribute to potential challenges whilst testing samples. I'm now going to hand you over to Hadas, my colleague, who has driven a number of key developmental areas within our laboratory, looking at the impacts of formalin treatments on various samples. And I think, as Joe mentioned at the start, if you do have any questions, please type away, and we'll get to them in 10 minutes or 15 minutes time, and then at the same time as poll. But without further ado, I'll hand you over to Hadas and her great data. Thank you.
Thank you, Jonathan. Hello, everybody. So today I would like to talk with you a little bit about how we manufacture our standards, and then really go deep into the data. And I would like to show you some data about our formalin-compromised DNA reference standards that we have produced. And hopefully you'll see how valuable they are in developing a new assay or assessing platform performance. So how do we actually manufacture our reference material? So at Horizon, we have developed a library of over 550 genetically defined cell lines, harboring mutations that can be found in cancer patients. We also developed their control cell lines from the same genetic background. These cell lines are extensively validated throughout a platform where our cell engineering team validates the cell line pre- and post-engineering. This includes a SNP6 assay to verify the cell line identity, standard sequencing to verify the accuracy of the engineered mutation or the gene editing, and digital PCR to assess the allelic frequencies and copy number of the mutation, and RT-PCR to analyze the gene expression. Our cell lines go through single cell dilution to ensure the clonality of the cell lines, and then we're working with highly pure homogeneous cell lines. By using these cell lines, these engineered cell lines, we produce a range of genetically defined reference standards ranging from DNA, RNA, FFPE sections, as well as cell lines on slides for fish and for IST. And altogether, these references are highly characterized, highly validated with a defined allelic frequencies of mutations. And now in my next slides, I will focus more on the formalin-compromised DNA reference standards that we have produced. Looking at DNA samples from a diagnostic lab, we can observe quite a lot of variability between samples. This variability at least partially originates from different formalin intensity levels, leading to differences in many aspects, for instance, fragmentation levels, the amplifiability of the DNA, as well as chemical modifications such as deamination and cross-linking. These characteristics can have a profound impact, as Jonathan mentioned, on clinical diagnostics and can lead to assay failure. Our formalin-compromised DNA reference standards mimic DNA derived from FFPE sections, using matched pairs of either formalin-treated or non-formalin-compromised DNA coming from the same batch of saline mixture allows us to produce reference materials with the same genetic background, with the same defined genetic allelic frequencies, but with only one aspect that is different, which is the formalin treatment. This can allow for a very powerful control to assess assay performance while taking into consideration the effects coming from the formalin. So let me start with a small example. So as Jonathan mentioned, DNA quantification is really important when you talk about how much DNA you would like to load on your assay downstream. So there are, of course, different ways to quantify the DNA, like the spectrophotometric base, like the nanodrop, the fluorescent base, like the QuantiFluor assay available from Promiga, or the qubit assay available from Invitrogen. There are also other assays like that are PCR based, like the qPCR or digital PCR, where you can back calculate the DNA amount you have based on the amplifiability. Now, for formalin-compromised DNA, we do not recommend using such ability, as we know that the amplifiability of the samples are reduced due to the effects coming from the formalin and the fragmentation. So here we focused on looking at what is the impact of formalin on DNA quantification. And if you look at the graph above here, we see several samples. Of course, this is just several samples taken from a very large study, where we quantified matched pairs of non-compromised on the top versus formalin-compromised DNA references on the bottom. And then we analyzed, or we measured the DNA quantity using the QuantiFluor assay, the Agilent tip station, and the nanodrop. And what I hope you can basically see is that there is a variation in the DNA concentration that is being measured by the different methods. The nanodrop seems to overestimate the DNA concentration. But if you look at the graph below, you see that the nanodrop overestimates, so that measurement, coming from the nanodrop overestimates the two other measurements when we're talking about formalin-compromised DNA. So to summarize, in formalin-compromised DNA, the nanodrop overestimates even more than in non-compromised DNA. Now, we've got also some data coming from the qubit assay, where we see the same consistent results, where the nanodrop, again, overestimates the DNA concentration over the qubit assay. Now, altogether, this is all very consistent with the previous paper just published a few weeks ago, showing how pre-PCR failures, including DNA quantification, can result in assay failure. And that was done specifically for the BRAF and EGFR mutations. If you would like to download this paper, it is publicly available for free.
Okay, and There should be a poll popping up on your screen right now, and we'll take a short break to answer any questions that you have popped into the box on the right-hand side.
Hi, so thank you very much for casting the vote on the poll. I'm not sure if you can see it up on your screen. In case you can't, there's it's very interesting. I think it's obviously a clear reflection of the popularity of this webinar. That if you combine the top two, the 67% of people find that formalin will have either a very high impact or a high impact on their assay, and 25 or 29% together is perhaps it won't have a great impact, but it's always interesting to get your opinion. So I appreciate your feedback, and I'll hand you back to Hannah to continue with the data.
Thank you, Jonathan. Thank you all for completing the poll. It's really interesting to see what you think. So just going back to the data, what I've showed you before is we've talked about DNA quantification and relationship to assay failure while taking into consideration formalin. And now let's move into looking into variant detection or mutant detection and what is the impact of formalin on that. So we have focused on the BRAF and EGFR loci because these two regions are known to be quite vulnerable to formalin fixation, probably due to the fact that these regions are of high structural complexity. So to assess the effect of formalin on assay performance and mutant detection in this loci, we have produced a range of BRAF V600E mutants with a range of allelic frequencies from 5% down to 0.2%. And we've prepared a matched pair of either formalin-treated or non-formalin-compromised DNA. So in blue you can see the mutant allelic frequencies detected by digital PCR on the non-compromised, and in red on the compromised DNA references. And what I hope you can appreciate is that we see an artificial slightly increase in digital PCR mutant detection due to the formalin. Then looking into another example for EGFR. So in this case, we have produced a range of wild-type EGFR reference material. So no EGFR T790M is expected to be found in these samples. Then what we've seen is that with increasing intensities of formalin, we've detected increasing allelic frequencies of artificial T790M coming up from probably due to the deamination on these loci due to the effects coming from the formalin. Now, this can result in non-specific amplification of this region, leading to an artificial mutant detection. What we've done with one of our very established customers is that we have provided them samples of EGFR T790M reference standards with a range of allelic frequencies from 3% down to 0.1%, as well as the control wild-type EGFR. And again, matched pairs of formalin, non-compromised and compromised DNA reference standards. And what was very nice is that we've provided the samples blinded for a customer who was interested in developing an EGFR assay, a specific assay for EGFR T790M. Now here you can see the results that our collaborators got, and then these results are based on 8 replicates, each for 2 independent samples. As you can see here, we got 2 samples with standard deviation and 8 replicates for each. And what was very nice about their results was that using these results, they could get full confidence in the specificity and the sensitivity of their assay. For instance, look that all the mutants were called as they should have, whereas in the wild type there was no mutant calling, which actually proves that they could get rid of the non-specific amplification that was associated with the digital PCR. What you can also see is that even with very low allelic frequencies and high intensity of formalin, they could recapitulate these results, which gives even extra confidence in their assay. Now, moving forward, we were also very much interested in producing a multiplex with, here, for instance, we have developed a multiplex with 11 validated mutations with a range of 25% down to 1% allelic frequency. And then we've also created a range of formalin intensity for this mixture. So basically, we have 3 different formats. One is the non-compromised, another is a medium intensity compromised DNA, and then the third is severely affected by formalin. What's nice about it is that recapitulates the variability that is found in DNA derived from FFPE samples, where you can see variability in the DNA fragmentation, in the fixation, etc. We have highly characterized our products, and for instance, here on the left, what you can see is that we've analyzed the DNA fragmentation of our products, and for one is the non-compromised DNA reference standard. You see it's high molecular weight, as we would assume, or as we would expect. And then number 2, you see we have fragmented DNA. And then number 3, with the severely compromised DNA, we see even more degraded DNA. Similarly, when you look at the DNA amplifiability, we've looked at the DNA amplifiability of our products using 2 different methods, the multiplex PCR and a qPCR, where we expect to get reduced amplifiability. And as we expect, number 1 is a DNA sample from non-compromised DNA. We see very nice amplifiable products up to 600 base pairs. Number 2, with a medium intensity formalin compromise, we see that there is a reduction in amplifiability. And number 3, with a severely formalin compromise, we see a massive reduction in amplifiability. And these standards are actually very interesting when you want to challenge your platform and see how good is your platform even when amplifiability is dramatically reduced. So to actually test one platform, we've got some very interesting data from one of our collaborators where they tested our material on an ion torrent platform with a custom panel. So what you see here, these are the 11 mutations I told you about with the expected allelic frequencies that were confirmed by us by digital PCR. And now here I'm showing you the data, the sequencing data that we've received from our collaborators. So on the left, you can see the data for the non-compromised DNA, and then HD7 150, the medium intensity compromised DNA, and lastly the severely intensity compromised DNA. And what you can basically see is that we're getting similar results in mutant callings throughout the products, which means that this workflow is very good at detecting the different mutants here in this panel. What you would also might see is that the EGFR T090M, which is 1%, which is the lowest one in this blend, was not called on that platform. It was not called on any of the products, so presumably there is an effect coming from the low allelic frequency of this mutant. What we cannot exclude is any extra bias coming from the formalin. And with this, I'm actually concluding this data set for now. Moving on to Jonathan.
Well, thank you, Hadass, for the excellent data you've presented to us today. What I'd like to do is bring it around to a full circle to where we were at the start of the webinar, and coming back to with the data, the work that we've done, and the samples we've started to develop to understand the impact of formalin, is bringing it around into actually being able to provide reference standards for you as a community to take this into your lab and start asking questions about your assay. And so what we've done historically, and it is important to mention that everything we provide is for research use only purposes. And what we've done historically is we have FFPE reference standards that are there to really test the whole workflow of your assay, because you would extract the DNA from the FFP, so you test how good your extraction is, how good the quantification is, and then can you detect the mutation or mutations on using your assay. And then we also have been providing DNA reference standards, which is very much focusing on the analytical part of the workflow, and can you detect, and can you actually then detect how can you actually analyze how good your assay is at testing for SNP. And what we're looking to do and taking Hadass's work forward is providing formalin-compromised DNA. And so what I've done here is try to provide a bit of a quadrant where, depending on the question you want to ask of your assay, what sample would be most useful to you. And it's always good as we go through product development that we get feedback from people like you who are experts in the lab, and you're running these types of assays day in, day out to provide where there may be gaps. And what we have is, if you're looking to test the limit of detection of your assay and the sensitivity of your assay, we have our DNA samples over here, which are very, very clean. They're straight from the cell lines. They haven't gone near formalin, so you're not necessarily testing the robustness, but the sensitivity. If you want to then start testing the robustness and the sensitivity of your assay, these are the samples that Hadass has been using in studies here. So this is a very severely formalin-compromised sample. This is the mild formalin-compromised sample, and that will test both the robustness and sensitivity of workflow. We then have, if you wanted to ask the question, well, what's the robustness and the sensitivity of your complete workflow, we can then provide the FFP sample, and then you extract the DNA from that and test for the specific mutations contained within the sample. And because of the way that we're approaching it at Horizon, all of these samples, the genotype of these samples are all identical. They all contain the basic 11 core mutations, primary mutations, and we know that the exact allelic frequency of all of those 11 core mutations. Behind that, we have an additional data for 29 secondary mutations. And we've actually done several next-generation sequencing runs. So we've got data sets of about 10,000 SNPs for the background cell lines we use, and so we can actually provide all that data for you. And so you know if you're going down the next-generation sequencing route, we can give you a lot of information up front to see, so you can really analyze what you should and what you shouldn't be looking for in your assay. And so that does bring this webinar around to a close. For me, the important part is to put the question back out there to you in that what would you like to test your assay for? Please do get in contact with us, because as we develop this as we're developing both on the research side as well as on the product side, we're very confident that we will be able to support you to test both the robustness and sensitivity of your assay as well as your workflow.